twist a little bit just to see whether you understand or not. So, but there is a reason why we're doing it, not just for the sake of manipulating questions to play with you. Okay, we are not playing anything with the students actually. There is a reason why we do it, because in the real situations, okay, when we do the research, and when you go to the labs and do research, remember, although if talking about research, the philosophy of research is something like we want to explore, we want to do so many things, but there is still limitations where the research are still funded. The research are funded, so we will have to work within the budgets. So in the real situations, sometimes we will have to work out within the budget, so that some research, although we know, sometimes we know the direction of research, and there's so many things that we can do, but sometimes we cannot do all because of the budget restrictions. So we have to manipulate a little bit so that we save cost. At the same time, the result that we generate are reasonable. At least something that are reasonable for publications. That's the end. At the end, we want to publish. And of course, it's not just for publication, but at the end, we want other colleagues to benefit from our publications. Let me show you the questions. Share. Okay, can you see? Can you see the questions? I'm sharing SAQ number 1. Okay, this is serial dilution. There are 2 stages where the dilution factor can be affected. There are 2 stages. First stages is the first serial dilutions where you transfer 1 ml to the different tubes. That's the first step. The second step is when you transfer on the plates. Okay, so these two stages are very important. If you do it wrong, it may affect the dilution factor. At the end, you are not, when you report the number of bacteria, it's not correct. So it is very important to ensure that the cell dilutions are correct, meaning that there is actually a series of steps when you do the cell dilutions. The easiest way is actually to keep with tenfold dilutions. The first step is normally we keep with tenfolds, meaning that if you add on 1 ml to 9 ml, it become 1/10. So that is 10 to the power of 1. That is the first dilutions. Then the next stage, you transfer another 1 ml. If you keep transferring 1 ml within the tenfold dilutions, then that will be very straightforward. It's easier. 10 to the power of 10, 1/10, which is 1 tenfold, 1/10, and then you transfer 1 ml to another tube, it will be 1/100, which is 10 to the power of 2, 10 to the power of 3, 10 to the power of 4. So easier. 10 to the power of 1, 10 to the power of 2, 10 to the power of 3, 10 to the power of 4. Okay, so 1, 2, 3, and 4, representing 10 to the power of 1 until 10 to the power of 4. Okay, the next stage, we want to transfer on the plates. Remember, the unit is CFU per mil, colony forming units. There is a reason why we use colony forming units, because we are counting the colonies. Although we are assuming that 1 colony raised from 1 single cell, that will depend on how good you're doing the spreadings. So if your spreading is good, you are able to spread all different cells to put a distance between one cell to another. So 1 cell will grow, become 1 colony. But there is still a chance, there is still a possibility that your spreading may not be good. There might be 2 cells still close to one another, and when they grow, the 2 will form a single colony. So that is possible, this is something which we perceive it may happen. That's why we have to minimize by doing good spreadings, we cannot guarantee that we will have 100% good quality when you do the spreading. It's impossible, but at least we can minimize. So that's why the units to compromise the potential mistake, we use CFU. We count the number of colonies. Although we are assuming that one colony raised from one single cell, but there might be a possibility that two cells may be close to one another. That at the end, when they grow, they form at the end they are all in a single colony, although originally they are coming from two or more cells. So that's why we use colony forming units per meals. Okay, so that's the tricks. The unit is per meals. So if you keep transferring 1 meals, okay, then the dilution factor will not be affected. But here, you are only when you transfer the volumes on the plates. Originally, you keep transferring 1 meals in the first serial dilutions. It's okay, very straightforward. You will get a good sequence of tenfold dilutions, starting from 10 to the power of 1 until 10 to the power of 4. But when you transfer on the plates, in these questions, you are only transferring 0.5. Okay, so if you are transferring 1 meals, then it's okay. We just simply find the one that fall within the range 30 to 300s. That is the rules. I think Dr. Azmiza already explained why we choose the one within 30 and 300, which is tip number 3. Okay, tip number 3. We have 200 CFU, while the rest are over the range. This is too much. Tip B, 2000, which is over 300, we just exclude. And the last one is about 10, which is too low. So we just exclude. The one that fall within the range is this one, tip number 3. So 200 CFU. This is the one that we take as the readings, but we have to multiply with the dilution factor. If you are transferring 1 meals, then we just straight away report as 200 times 10 power of the dilution factor of the earlier tubes, which is 10 power of 3. So 200 times 10 power of 3. Very straightforward. Provided if you are transferring 1 meals, because you are reporting as CFU per meals. But here, you are only transferring 0.5, not 1 meals. Okay, so you will have to do extra calculation so that at the end it will be normalized to become per meals. Okay, so that is a trick, and the trick is not just to manipulate the questions, but there is a reason why we're doing it, because in the labs, although there are different sizes of plates, we have a small plates, a standard size, split of standard size, or we can have a bigger plates. But we don't buy so many different size of plates. It's just a waste of, that will add on cost. We just buy one size, one single size, it save costs. That's a reason. As I said earlier, we have to work within a budget. So we don't waste extra money for something which is not necessary, because we can just stick to one size and we save a lot of money rather than buying different size, because there will end up, you paying more. Perhaps the smaller plate may cost you more because it's not easy to manufacture small plates. Okay, in the lab that we have, the plate that we have in the lab is the standard plate, which is about, what's the diameter? It's about 24, it's about 20 cm. Check. I have the ruler here. Oh, sorry. It's about 8 to 9 cm, the size. Okay, what's the problem with this size? It's no problem actually, but the problem here is based on our experience. If you want to transfer 1 meals, it will take some time for the suspension to dry, because 1 meal is quite, the volume is quite huge. It may take some time to dry, which is we try to avoid. So normally it works best to use 100 microliters, less than 1 meals. Sometimes we use 100 microliters only. We just transfer 100 microliters so that you can spread easily and it will dry faster using the standard size. Of course, if you have a larger size, you can transfer 1 meal, 2 meals, because plates is big. But the one that we have in the lab is just, normally we keep working with a single standard size, which is about 8 to centimeter in size. So there is a reason. This is not just a trick, but there is a reason behind this. So here we put you in the question. We put it here like 0.5, less than 1 mil. You just transfer 0.5. But the thing is, when you transfer less than 1 mil, while at the end you want to report as per mils, definitely the dilution factor will be affected. So to report the reading is actually, first, of course, you will have to pick up the right one, which is 200s, plate number plate number 3, which has 200 colonies, which is within the range. Then multiply by the dilution factor, which is 10 to the power of 3. But because of the smaller amount that you transfer on the plate, which is less than 1 mil, you have to do extra calculation, which is you have to divide by 0.5. Okay? You have to divide by 0.5 first, then you can report as CFU per mil. So the result will be about 200 divided by 0.5. It will be how much? How many? 4. The answer is 4 times 10 to the power 5. That is the correct answer. Okay. Some of you, you pick the correct place, so you got partial mark for that. But when you report the final readings, you forget about the 0.5 things, so you lose mark from that. Okay. So, but a few of you did well. You did calculate properly and get the actual result, which is about 4 times 10 to the power 5. Okay, so that's a trick. Of course, this is probably a bit mathematics, but this is very important techniques in microbiology. We need to dilute, because microbes, if you provide the proper environments, proper media, they will grow, and they will, one cell will become 2 within a day. Within overnight, you will get thousands. So, which is a bit difficult for us to deal with 1,000 cells using our naked eyes, unless you use other method like ODs. If you measure the OD, probably that would not be an issue. But if you want to count the colonies, then you have to work within the capacity of your eyes. So that's why we have to dilute. But of course, when you do the dilution, you have to monitor the dilution, so that at the end you are reporting the correct numbers that will represent the actual number in original suspensions. The other thing is for SQ number 2, all of you, majority of you did answer well, except that you are not being concise enough. Okay, as a scientist, we have to be very concise, very clear. I think all of you understand what you mean by aseptics, and you know, because you have done this in your practicals, you know, such as wearing gloves, sterilize using the 70% alcohols, work close to the Bunsen burner, to the heat source, because the heat source will carry all the hot air will carry away from your working area. So somehow, if there is airborne contaminants, those contaminants will be carried away. So that will minimize the contaminations. I think you understand, because many of you just simply put all these points. Why? Because I also asked for the reason why, to minimize contamination, which is true, but it's not concise enough. But some of you did very well. They explain further. Okay, to kill, okay, or to remove all the potential contaminants that are present in the surroundings. That is more concise. It reflects better. Okay, rather than just simply by saying I burnt my wire loop to sterilize, which is okay, it's true. But why? To sterilize, what happened when you sterilize? Why you want to sterilize? Because there are contaminants on the wire loops, so you want to heat up to kill. It fulfills the definition of sterilizations, but when you simply put sterilize, it's quite broad. So you have to be more concise by saying exactly what's actually going on. So those who being concise, they got full marks, but those who just simply put like just to sterilize, then they got partial marks. Okay, so I hope you learned. This is I know this is actually the skills in answering questions. Although you understand what's going on, but you have to practice yourself to be more descriptive, because that is the nature of being a scientist. That makes you different from the other programs. Biomedical scientists, you will be doing a lot of work in the labs. So this is the culture, the way you provide information, the way you explain, the way you present will have to be very concise to reflect actually what's going on, rather than just simply to put general terms. Okay. So hopefully in the second test, you learn something from here. Hopefully in the next examination, test 2, which is around, what's the date? What's the, I have to adjust. If you look at the timetable, the original dates is end of May, which is, I think, is too early, because we just finished our test, our first exams. So if you want to do it in the final week of May, that will be about 3 more weeks, which is, I think, quite near, quite short. So I'm going to move the second test in June, early of June, probably in week 10. You might want to double check your timetables. The test 2 will be in June, the first week of June, or in week 10 or week 11, but most likely it will be in week 10, which I already checked the dates. It should be early of June. Same venue, same times. We will utilize our practical science to do the test 2. Okay, so get ready, and test 2 will cover my parts and a little bit on the practicals from your same things, the same questions, the practical questions. And also for my part, and also Dr. Mila. So I will provide more info on that later on. Okay, so any questions on the test 1? No questions? Let me, okay. This is your results in your Petroblast. You can go and check. And then by end of this week, I will key in SMP, and you can check the marks in SMP whether it's consistent with the one that I have here. Just in case if sometimes we did a mistake, a lot of numbers to key in. Okay, and then so this is the topic that I'm going to talk about. These files will cover all my lectures. Okay, I have 3 to 4 sessions for my lectures. So this slide will cover everything. And at the same time, I also provide reading supplements to enhance your understandings. Okay. So if you have time, you can go through. And I might want to discuss on this later on. You can look at the learning outcomes. For each of these reading supplements, there are learning outcomes. So the expectations, what we expect you to understand from these contents. So you might want to see what are the expectations so that you know the scopes. Okay, sometimes actually what we are learning from this course is very basic, a lot more, which is we don't. One course is not enough to learn about microbes. My BSc, my bachelor in microbiology. So I studied microbiology in the beginnings. From my bachelor, I'm majoring in microbiology, and then I did my master and PhD also in bacteriology. So in my programs, actually what you studied in this course is actually it's cover everything, but crammed in a single course. So actually the topic that I'm going to deliver today is actually coming from a number of courses from my programs. There is actually a course for microbial physiology. There is a separate course for microbial genetics. There is a separate course for pathogenesis, bacterial pathogenesis, and different course for viral pathogenesis, fungal pathogenesis. So that will take up 4 years, while everything's being crammed in a single course for you. So, but it's okay, because you are not majoring in microbiology, you are studying biomedical science, which is very broad. Not to mention microbiology is one of the biomedical science courses, but there are other courses like pharmacology, molecular biology, so it's too huge. But I believe there's some good aspect on this because I don't believe that a bachelor is everything. So, bachelor program is not everything. You may specialize a little bit, but it should be, I don't know, because I studied in the United States, the approach is a bit different. We got more time to explore. While in Malaysia, the program designed to cater for the industry. We want to produce someone who can work immediately. When you finish your study, you can go straight away. That will depend on the country, yeah, because we are still a third world country. You cannot compare with the first world countries because they have advancings a lot more than us. And in fact, when they are too advanced, sometimes they cannot practice in Malaysia because our environment is different. The medical doctors graduated from US cannot practice in Malaysia because it's just a different environments. Although they are good, but we don't have all the technologies where they can practice. They are familiar with all the systems, with all technologies, with all the supporting systems, but we don't have it here. So to work in Malaysia, we have to go through our own systems. So that's why when we want to send students overseas to study medicines, we send to countries where the systems are similar to us. For example, we send to Indonesia, we send to Egypt, we send to Bangladesh, India, because the scenario is a bit similar with Malaysia. So we have more hands-on. But in US, I think it's a bit less on the hands-on aspect because they got systems where they can't simply touch the patients without permissions. So somehow there is not a practice here. I'm not saying that we are better or they are better, but it's just simply that we are working in different environment that we need a special, if you want to train doctors to work in Malaysia, we have to train according to our priorities. Okay, so the doctors graduate from the United States, they cannot practice in Malaysia, although they are more advanced. And of course, our doctors may not be recognized to work in the US. That one I'm not sure. But what I'm sure is that doctors from the United States, they can practice in Malaysia. It's just totally a different environment. So that's why when we design the course, okay, we have to cram everything. So we just want to, this is more like an exposure for you to know the priorities. Okay, microbiology is very huge subjects. Okay, what we are studying here, this is medical microbiology, just a small part of the whole scopes, because only a proportion of microbes are actually causing disease. Majority are actually good microbes, and in fact, they are part of the ecosystems. We are relying on microbes to sustain the ecosystems, and we also rely on microbes to sustain our economy, to sustain the industries and foods, okay, as well as medicines. Why? Because in medicines, normally, if you're talking about medical microbiology, it's all about identifications. We want to know exactly what is the causing organism so that we are clear what to do. The next thing is to know what is the antimicrobial susceptibility patterns. Okay, we'll be doing this in your practical this week. After you isolate and identify, then you want to find out what are the antimicrobial resistant patterns. We want to know whether they are resistant to what and they are susceptible to what, so that we can prescribe the proper antibiotics. Okay, and then we want to come up with preventions. Normally, if you're talking about microbes, you will be dealing with the vaccinations. Okay, that's the way to to induce your immunes to deal with a particular group of microbes. So basically, it's all about isolations, antimicrobial susceptibility patterns, and preventions, which involve vaccinations, controls. There could be other means of control. Vaccination is important, but what are the other means of control? Biological vector, that probably you will learn more in the other course. Parasitology course, probably they're talking about the vector bonds, pathogens where, for example, for dengue, it's a virus, but to deal with dengue, we have to deal with the mosquito. okay, the vectors that are carrying the viruses. Okay, so, but in this course, we just focus on the bacterial disease, viral disease, the most common ones. So basically, as far as the routine practices are concerned, it's all about, when you work in the university lab, it's all about identifications, the microbial susceptibility assay determines the range of antibiotics which are commonly being used to treat the particular microbes, and something to do with the prevention. But if you want to do research, there are a lot more. Okay, as far as the medical bacteriology concerns, you have to understand the pathogenesis to find out where you can intervene. If you want to design the vaccines, if you want to design an alternative for antibiotics, because now we're dealing with the emergence of resistance. So is there any other compounds that might work? There are, but still need a lot of research. What are they? Apart from antibiotics, previously, before the antibiotics are available, we are relying on the human civilization at the time, they are relying on chemicals. Okay? For example, those chemicals that are used to treat syphilis and so on. And of course, chemical, they come with the side effects. And later on, when they discovered penicillins, the first antibiotic, then they somehow that trigger them. If there is an penicillin was the first antibiotics. If there is an antibiotics, perhaps there are a lot more compounds, antimicrobial compounds. Okay, we have found one, penicillins, but there could be more. So then they began to explore and they found more. Okay, starting from penicillins, then there are what else? Erythromycins, macrolides, and so on. A lot more. We are not going to discuss this. This is basic microbiology. Probably we will discuss this in the advanced microbiology. You will be taking advanced microbes in your third year. You will be seeing again to discuss on these issues. And then what else? To understand the disease developments, how the microbe interact with our whole systems, and how to evade our immune. So we want to intervene somewhere. Okay, so that is the research. If you're really interested to do research, that's just within the scope of medicines. But as I said earlier, only small proportions of microbes are causing disease. Okay, the majority are actually good microbes. So that's why we have industrial microbiology, food microbiology, environmental microbiology. Okay, so a lot more microbes which are actually, if you are studying if you're looking into compounds to treat microbes, antibiotics, and antibiotics are produced by microbes. So some group of microbes are producing good things, good products that we can use to treat other dangerous microbes. Not only to treat other dangerous microbes, but some compounds are also useful in our food preparations, food preservations, and food productions, because sometimes we use enzymes in our cooking to prepare certain food. We need enzyme, and these enzymes are coming from microbes. So this is food microbiology, and of course there are a lot more kind of enzymes which are not only for food but also useful for synthesis of certain things. So we have environmental microbiology. Okay, so it's a huge scope. So that's what I learned from my undergraduate, because I'm majoring in microbiology. But now we just want to focus within the scope of medical microbiology, which is just a small part of it actually. But it's important because people die every day because of the infectious disease. So although it's only involved a small proportion of microbes, but the problem is still persisting until now. And perhaps in the future, if we don't do something, probably it may get worse. Now we are probably recovering from COVID. So which I think there is no such thing as recovering. It's just slowing down a little bit, but in the next few years, probably there will be new emergence. So it's not recovering actually, just put on silent for a while before the microbes emerge and cause a new pandemics. We have seen in history, in the past, there has been several outbreaks. So we did learn actually. So that's why for this course, we include history. Okay, so whether you like it or not, that's in my programs. there is one course where we studied history. Why? Because we want to see what's going on in the past. Okay, we want to learn from the past, and it's very good for us to guide us the direction for how to proceed towards the future. Okay, normally we look at the past to see the patterns, a lot of things to learn, so that those will guide us how to move forwards. One thing that we learned is actually we have to keep exploring. Okay, we have established a collection of microbes. Okay, we know there are E. coli, Salmonella, Shigella, but actually, if we keep exploring, we will find more new species, perhaps. So what we have now, the range of media that we have now are actually established to deal with the microbe that we have recognized. So how about microbe that we have yet to recognize? Do we have a proper media for them? Probably we don't discover because our media are not formulated to harbor or to allow this new microbe to grow. They work well with the microbe that we have recognized, okay, E. coli and so on. These are already established and we have a proper media for them. But probably these media are not good enough to catch more new potential species. So perhaps we have to come up with a new formulation of media to harvest more. So that is one of the things that we learned, that we have to keep exploring. So although it sounds waste of money, why you want to keep something which there is no guarantee that's true. Sometimes we have to balance between the priority or economy, at the same time to explore new things. So you have to be balanced. Okay, that's just introductions. Now we go to our slides. I'm going to share again what I'm already sharing now. Okay, this is the first slide. We have seen this. I'm going to skip the slide. This is just I already discussed this earlier in the earlier semester. You have seen this. You have done this in your practicals. You have done streakings. You have seen the media. You have seen the colonies, and you have seen the single cells. You have done staining to see the cellular shapes, gram positive, gram negative. Those are very basics. So this technique has been there since long time ago. Okay, the first, I'm not going to talk on the history yet. That will be next week. Today, I'm going to discuss the basic characteristics. But sometimes we have to go back to the history to, to point out the importance of this. So the streakings, the agar media, the plates has been invented long time ago, since the beginnings. At that time, we call when we look at, the time frames, we can classify according to the advancements, starting from the time where we do nothing. We call it time where we know nothing. Then come with the golden age, when we realize the existence of microbe. Then that trigger researchers to carry out more research and establish some information and some basic techniques. That comes in technique such as streaking and so on. So that is the beginning of the golden age. That's what they call. Then come the modern age. Now we're talking about, now we are living in what centuries now? We are talking about very specialized aspects of microbes, starting from the genomics. When the first group of scientists discovered the existence of DNA, the genetic informations, then the first models where humans began to realize the existence of DNA is microbes. Okay, we have DNA, but the DNA was first discovered in microbes. Then, of course, because microbes are living cells, then we are also made up from living cells. So now we know that our cells also have DNA. Okay, so starting from the microbe as a models. Okay, and then now we're talking about when they discover the existence of DNA, then they're talking about the whole genetic makeups, the genomics. So we have seen the genomic era, the advancement in the genomic era that come with the technologies, because we want to deal with it, we need tools. And the tools starting from wire loops, That's among the primitive tools which we are still using it today because it's very practical, very cheap, very practical. But at the same time, they come up with the new tools. At the same time, concurrently the development in the IT, in the computers, where we can use it to help us, to deal with this because it's too huge. So we need the technology to help us. So the genomic come with the technologies around the computer systems, okay, the bioinformatics. And then, of course, from the genomics, that is just a DNA, genetic information, but how we want to understand the expressions, the protein level. Proteins from the primary sequence, just a piece of amino acid sequence, being folded to form a secondary, and then being further folded to form a tertiary. The tertiary structure is the functioning proteins. Okay, starting from a single piece, they're being folded in a certain way that will determine the specific functions, the functioning, the tertiary structures. Okay, the enzyme and so on, the working enzyme are actually proteins being folded in a certain way that carry certain functions. So after genomic, we have a proteomics. Now this is about integration between genomic and proteomics, and blah, blah, blah. I'm not going to discuss that, that will take too long. That is actually the transition, the advancement in microbiology. We started with microbe. Why? Because it's the simple things, the simple living things. That's why in this course, we did compare between microbes and the higher and the more complex cells, which is, we have actually terms to refer this, the prokaryotes and eukaryotes. Okay? Prokaryotes, the unicellular, single cells. Eukaryote is actually a multi-cells, multicellular levels, which is our bodies. Our bodies are made up from a single cell, but starting from a single cell, which is, it cannot function, one cell cannot function, it requires other cells, because we are multi-purpose, we can do many things. So that is actually because of a combination of many cells that have a different functions. So we are eukaryotes, while microbe is a prokaryote, a single cells. They are carrying just a basic things, but they have caused a lot of problems. Because they're very small, they can enter our bodies and interfere with our systems that lead to disease. So that's a problem. So basically, to simplify everything, because it's only one course to cover everything. So basically, the major categories that I want to emphasize for the course, there are only three. There are more than three actually, but these are the three most dominant groups that are common in causing diseases: bacteria, fungus, and viruses. Among these three, we will be talking more on bacteria and virus. Fungus do cause disease, but in our country, it's not as aggressive, not much serious as bacterial and virus infections. So sometimes we have to, the way we deliver things is actually to accommodate, to be consistent with what we have in our country. So we are dealing a lot more with bacterial and viral infections, which is true in many other countries as well. But if you go to the poor country, probably some remote country in Africa, probably you might want to learn more about fungus because of the poor hygiene and so on. So although we don't have so many serious cases involving fungal infection, but in other countries where they have a poor sanitation system, poor hygiene practice and so on, probably the fungus, we need to know more about fungus, because fungus are, although not common here, but they are common in other countries. There are other groups like amoeba, those that we never heard of. We don't know whether people die in Malaysia because of amoeba infections, but in other countries it happened. That will depend on how good is the socioeconomics of a certain country. So sometimes we have to, to select what are the subjects that are relevant to our countries, because we are here. But we do have international students that, of course, this is good for you to see, the differences, how the differences affects the health quality. Okay, so that probably we will not discuss this. It's too huge. We are going to just to look at on the basic characteristics, so that it will serve as a fundamentals. Okay, for you. Later on, you can move on. Okay, to look more, to go into, to explore more. The Okay. So these are the scope which are relevant to microorganisms, foods, ecosystems, beneficial products such as antibiotics, the research tools. Don't forget, okay, to deal with micro, we need to invent tools. Okay, without tool, then we cannot do anything. You are familiar, you have gone to the labs, you have seen the basic tools. That is just the basics, a lot more. If you go to the research lab, probably you can find more. In fact, you can see highly sophisticated big tools. Why it looks just piece of wire, but if you go to the virology labs, you can see the safety cabinets, you can see a huge CO2 incubators, okay? Incubator that have to be supplied with the CO2 because that's the nature of the virus. Actually, it's not to grow virus. It actually is to grow host cells. Okay, because we need the host cell to infect with the virus because virus cannot be cultured on medium because they are not a living entity. Bacteria is a living entity. It can stand on its own. It's a living cells. Virus is not a cells, although it's a cells in the sense that there is actually a structure, but the structure is not carrying out metabolic reaction. It's just a piece of DNA or RNA being encapsulated with the capsids. That's it. Okay. But once it enters living cells, our machinery, our cellular machinery can recognize that particular piece of information where it will translate that information. And at the end, the information is actually leading to the formation of new viruses. Then there will be accumulation of the new viruses. At the end, the cell will explode. And then the new particle will go and to the nearby cell, and the same process will keep repeating. So at the end, that will lead to some pathological complications. Our cells are being damaged and so on. And at the same time, our our brain will will send a message to send the immune cell to come and try to control it. And that will lead to the feelings, pain, and so on. So definitely there will be a level of damages that may be permanent. Okay, so the impact is very huge. Although it's starting from just a single piece of information, but if we don't do something, that will lead to something serious at the end. And people die because of the viral infections. Okay, these are some of the disease. Okay, bad or good? That is very subjective. Bad or good? There are bad microbes, there are good microbes. And so what shall we do? What can we do just to understand them so that we can minimize the bad, so that we can minimize the adverse effects of the micro-infections, okay? And it's good to, somehow at the same time, it will create knowledge, and knowledge will require a group of people, and these people will require to be trained and exploring. So this is somehow lead to the career developments. So we are here because of microbes. We have doctors because of microbes. If we don't have microbes, probably we don't need the doctors. So in a way, it's a balance. It helps to stimulate the career, the jobs, opportunities, and so on. At the same time, we have to do something not to, at least to balance. We will have disease. That's just normal. But we want to keep it minimum. Okay. Okay, so now we start with the very basics. How do we classify? I already mentioned we have prokaryote, eukaryotes. So we want to make some comparison so that we understand. In the complex, at the end, it's all about interaction between microbes and the host cells. Okay, so that's why we have to know some differences. What are the basic differences? So prokaryotes, how they differ from eukaryotes. So we will look at the size, cellular structure, and arrangement of the DNA. There are obvious differences between prokaryote and eukaryotes. And then, of course, there are very unique characteristics related to the microbes. They're very small. They're living unicellular cells. They're very small, which is beyond the resolution of our eyes. Normally, it's below 100 micrometers, which is beyond the capacity of our eyes. Our eyes can recognize, if you have a bigger resolution, more than 100, probably our eyes can, you can see it, with our naked eyes. But below than that, we don't have the capacity. But we can use, we have tools to help us, which is microscopes. Okay, so there is tools. Okay, so now you can see the correlation between tools, the biology, and the technology. And technology is all about inventions of the tools to help us to study the biological phenomena. While eukaryotes, we are eukaryotes, we can see the size of our cells are bigger, and the arrangements of the DNA are a bit different. Okay, this is the size. If you put on scales, you can see these are living creatures, complex microorganisms, but still considered under the microbiology because they're very small. But they are big complex. Then we go down, you can see our individual cells, such as white blood cell, red blood cells. And then we have to go down further to see the size of the microbes. So microbes are located within somewhere here. Okay, this is our cells, bigger. This is our cellular components. In our cells, there are organelles, separate organelles, compartmentalized structure. Okay, so one of the is mitochondria. This is mitochondria. One of the organelles is mitochondria, which is part of our cells. The function is actually to generate energy. And then the bacteria is smaller than our cellular components. Okay, this is our cell, this is our cellular components, this is microbes. And viruses are a lot smaller. And then come the basic chemical structure, the lipids, what else? The protein and so on. Those are the basic elements. The lipids, proteins, DNAs, carbohydrates. Those are the basic chemical elements that constitute cells, the living cells, that constitute bacterial cells, that constitute our cell, and our cell also constitute aggregate to become a higher organelles that carry many various functions. So what are the implications of this? Why we want to know this? There's a reason, actually, not just for the sake of knowledge, but we can see on the application aspects. As we know, viruses are smaller than bacteria, and bacteria are smaller than cellular organelles, which constitute the higher cells of human cell, for example. So meaning that bacteria may be invaded by virus as well. So not only humans are being subjected to bacterial infections or viral infection, but bacteria are also subjected to viral infections because viruses are very smaller, are a lot more smaller. And in fact, bacteria, there are specific viruses that invade specific bacteria. So the virus that invade bacteria, we call a phage virus that affect bacteria. So they are also being invaded by the virus. So they are not safe as well, as far as we are concerned about being infected by bacteria, but bacteria also being infected by the virus. And of course, definitely, we are subjected to viral bacterial infections. There has been a theory, mitochondria, it looks similar. If you look at the shapes, that look similar with the bacteria. There has been a theory that proposed that actually the mitochondria is originally a bacteria that somehow integrate with the human cells and assimilated and become part of the cells. That's just a theory, but makes sense at a certain extent, because organelles, mitochondria, somehow they show the characteristics of being independent cells because they have their own DNA. Okay, they can carry out the function of mitochondria is actually to generate energy. Energy is required by the whole cells to sustain the system. You need energy, chemical energy. If you can recall from your high school, ATP, adenosine triphosphates, by losing there are three components. By losing one, that will generate spark energy. Then they reform to form the three molecule will reform, and then during the process will lose one component. During the process, energy will be released. We are not talking, if you was, I studied that, we call it, there is actually a cycle, the sugar cycles. How many energy being generated from a single molecule, from a single sugar molecules, it will be a single sugar molecule, it will be degraded into a simpler forms. A single complex sugar molecules will be degraded to become simpler forms. During the process, energy will be released, and the simpler form that are being released will be utilized to resynthesize a new structure. Okay, so it's all about, actually we don't lose anything. It's all about transformation from one structure to another, and one simple structure will go back and form a complex structure, and during the process, energy will be released. So that is actually very basics. So I'm saying this, we want to understand what happens when we eat food, when we eat some things. Okay, what happened in your stomach? It will be degraded, okay, into simpler forms. The simpler forms may consist intermediary simple components that will be utilized to form a new structure, to form your organ and so on. One of the structure is your chromosomes, your DNAs. Okay, your chromosome is actually, it contains DNA, but a very compact sequence of DNA. But if you're talking about a single component of the DNA, there are 4 bases of DNA. Adenosines, what else? There are 4. I mean, they are being sequenced to form a chromosomes, carrying a specific informations. But where do the single form coming from? The single adenosine, where do they coming from? From your foods. Okay, your foods is a complex structure which contain DNA and everything. And when you go to your stomach, you will be digested, you will be degraded, and in the chromosomes, the complex structure of DNA in the food will be degraded into individual components, and that individual component will be utilized to form your chromosomes, okay, to form a new cell which contain DNAs. Okay, so this is just a cycle, and energy will be released. So same thing with bacteria, they have to feed, to look for this supply, and they grow very fast. We don't grow as fast as bacteria, but bacteria multiply every 30 minutes, the fastest rate, which the characteristic of E. coli is very well studied organisms. So every 30 minutes, one cell will become 2. So to form a new cells, you require all the supplies, all the materials. Where does the material come from? That's why the bacteria need to feed on foods. And we are the foods, okay? They feed on us to get all the supplies. So they are just, make the livings. But of course, it can lead to something else, okay? It can be some adverse effects, lead to disease, but they are doing this not because they are very bad. They're not bad, actually, they don't know. They just want to make the livings. So once they find a suitable environments where they can get all material, then they just simply, utilize the surroundings. So don't blame the microbes, because they just make a livings. And we humans who have the capacity to think, so we have to accept that this is the reality of life, but at least what we can do, we can minimize. We cannot, it's impossible to wipe completely out all these potential risks. It's possible, but what we can do is just to minimize and try to sustain life. Probably we can live a bit longer. One day we're going to die, no issues. Everyone will die, but at least we die peacefully. We live longer with full satisfactions. Okay, so this is the inside of the cells. Okay, this is the eukaryotes and this is the prokaryotes. There's a huge difference. You can see what a difference. Probably the figure does not really reflects because everything has been put up on screens, simplified. But actually, you can notice look at the organelles. There are Okay, if you pick one eukaryotic cells, okay, if you can go inside and see what's there, although it's not impossible, but we do have technologies. Again, the technologies we have. What are the technologies? We have scanning electron microscopes. Okay, so that can penetrate something like a beams. I don't know. I'm not a technologist. Somehow that will reflect, give you the internal structure. Same thing where we measure the depths of the oceans. We don't actually dive to the bottom of the oceans. We just send us beam signals or electromagnetic wave. Okay, then it will go deeps. Okay, and then will be reflected back and being picked up by the machines. So that's how we know how deep they are, by based on the how long the beam will take and being reflected back to the machines. Same thing here in electron microscope. They just simply send a wave, and it's a bit complicated to explain, but somehow it can give some reflections on what's actually the environment inside the cell. And definitely, you can see this is very obvious. You can see there are compartmentalized organelles, and when you look at in the prokaryotic cell, there is nothing except the DNAs. In fact, the DNA are floating, not in the eukaryotic cell. The DNA are being encapsulated in the nucleus. There is a structure. There's another cellular structure we call nucleus, where it contains the chromosomes. Which is the DNA. Yeah? So while in the prokaryotic cell, DNA are there, but they are floating. There is no specific organelle to encapsulate the DNA. It's just simply DNA being floating in the cytoplasm, in the suspension. Okay, this is a nucleus, a chromosome, and then this is a bacterial cells with the DNA floating in the cytoplasms. Okay, now how the DNA being translated into proteins, into functional proteins. Okay, that's why bacteria serve as the first model to understand the DNA, because it's very simple. Okay, in bacterial cells, everything is taking place concurrently in the cytoplasm. While in the eukaryotic cells, there is a specialized organelle that carry out certain activities. One obvious activities are the DNA replications and DNA transcriptions and DNA translations. Okay? In the eukaryotic cells, DNA are being inside the nucleus, and then it will have to be translated into messenger RNA, and the messenger RNA will have to be taken out, will have to be exported out from the nucleus, where there is another machineries waiting here to translate the mRNA into functioning proteins, into proteins. Then the protein will be further folded in the tertiary structure to be a functioning proteins. Okay, the activities are located here, not inside. Inside is just DNA and mRNA translation. While in the prokaryotic cells, everything is taking place at the same time. Okay, so there will be a traffic jams. But so it's very efficient. This is a very wonderful phenomenon. We are expecting a traffic jams because there is no specific organelle to separate all activities. Everything is taking place in single, at the same times. So how would that particular information be synthesized without being, sometimes overcrossed with some other, I cannot imagine, but it's happening efficiently. So this is something very wonderful. God has created this very wonderful. So while in humans, we can see some segregations of activities. Now, what would these be the implications? Okay, of course, I always ask this. Why? Because we want to see what's actually, so what's the problem? If you know this, then what would be the benefits, or how the effects, us as humans? Probably the best example now, since we are since we are still struggling with the COVID pandemics. So now we have mRNA vaccines. Okay, so how we want to ensure it's safe? mRNA. People are complaining that mRNA are not safe because it can get into the nucleus. You can get into the nucleus and integrate into the DNA, and you will be somebody else due to the mutations, which makes sense at a certain extent, but we have to be very careful to see what exactly actually the system is all about. So mRNA is a piece of information. Once we inject into the cells, when we got into the cell, it will be translated. Same thing as a virus. Viruses are carrying genetic information. When it got into these cells, the cell machinery will translate it. Okay? But I would believe that there may not be the possibility of the DNA vaccines, the mRNA vaccines. We can see here, mRNA are being synthesized here and being exported out. Once it's exported out, then it will be translated by ribosomes, another organelle that are synthesizing DNA to form proteins. So if you're using the mRNA vaccines, probably it will just be around this area. It will not get into the nucleus. So most likely it will not affect our chromosomes. It will not get into. What happened here is the exportation. What happened here is actually our mRNA being exported into the cytoplasm, not the other way around. Unless if you can prove, I don't know, in the future, probably we have new technologies to prove that actually mRNA can get back into the nucleus. I don't know. But what we know now is actually, it's always being, the case is actually, it's always about mRNA being exported. So we may get the mRNA vaccine, it will be here. And once it got to the cell, it will be synthesized by the machinery system into proteins. And the proteins is a good antigenic molecule to induce antibodies. Okay, so that is the rationale of using mRNA. And of course, we have to investigate further to see whether it's really, because we are dealing with the humans' diversity. Because humans are diverse, although we look similar, but they're at a micro level, we are different. That's why we have certain people are more prone to a certain disease because they are carrying, we have genes, but we have a different polymorphisms. So some are lacking a certain, some are carrying certain more polymorphism that are prone to certain disease, for example, diabetic and so on. Okay? That's why diabetics, although we're talking about don't eat too much sugar, but sometimes there is some inheritance factor because of your polymorphisms. So this is just to throw something for you to think about. You can learn in other courses, probably Dr. Shwali, Dr. Prof. Cherkin, they may be in their course, they may be elaborating more on this, but we want to confine to microbes. Okay, another difference is that in the microbes, bacteria, a piece of information is already complete, carrying a certain information. For example, gene A is carrying a certain information for proteins. It's complete from the beginning to the end. So normally, for one particular information, you will have the start codons. Okay, it requires enzyme to come and initiate the synthesis. So what are the enzymes? The Taq polymerase and so on. Taq polymerase, I mentioned this because we are going to use it in the next practicals. Taq polymerase is enzyme that do the DNA replication. Okay, so we are going to use these enzymes to amplify DNA in your next practical. Okay, that's one of the enzymes. That's one of the enzymes. A lot more enzymes. So you will go and find, you will have to find the starting points. Okay, so there is actually something like sensing the DNA sequence that are actually encoding to serve at a starting point. So the machinery will recognize this as a starting point and then start the amplifications. Where to stop? Okay. There is another sensing, another sequence that are actually encoding a stop point. Okay. At the end, at the one end of the at the other ends of the DNA sequence. So it will stop at that point. So that piece of fragment will be translated into proteins and form a certain functions. But that is not the case in the human cells. Okay, it's not continuous. there are some interruptings, somehow like a short fragment that are actually uh, uh, although if you take a long piece of DNA, actually that is supposed to encode a certain functions. But along the sequence, there are interrupting sequence. We call it codons. We call it the introns. Okay, introns. Introns. So this intron will have to be removed. So it's not straightforward. Starting from the long piece of DNA, a piece, one segment is supposed to encode a certain functions, but there is another step that will have to be formed here, which is to remove all these interrupting sequence, the intron, so that what left are the exons, the actual encoding sequence. And of course, because they are being segmented, there will have to be, after intron will remove, then all these fragmented exons will have to be combined to form a single piece before they are ready to be translated into messenger RNA. And messenger RNA will be transported out from this. And outside, there are other machinery waiting, the ribosome, to translate the messenger RNA into proteins. Okay, so these are the summary. I want you to look at this table to see the differences between bacteria and the higher groups. Okay, what are the differences? There are other differences which we don't have much time to discuss. Okay. Okay. Now this is the very unique characteristic of microbes. Okay. Among the earlier approach since the beginning, from the beginning era of the modern microbiology, this is the first technique that being developed by utilizing the cell walls to differentiate. Okay. We found out that because the problem is microbe very small, we can't see. So we have to have tools to grow them. We have to provide media. Once they grow, we found out that they are expressing some sort of, a different morphological looks and so on. So that's a big challenge. How do we know the one that are actually relevant to disease? How do we differentiate those that are good microbes from the bad microbes? So unfortunately, when we're talking about those that are causing disease, there are not so much difference. On plates, they don't have much difference. They all look similar. Creamy, yellowish, a bit yellowish, whitish, creamy, but not much different to else. So how we want to differentiate them? But as far as the other microbes, if you're talking about environmental microbiology, where we're dealing with fungus-like bacteria, for example, Streptomyces, they're very colorful. If in fact fungus, okay, Dr. Wesley will be talking more on fungus. They're very colorful, where we can make use of these traits. We can make use of these morphological looks to differentiate them into species, into groups. We're not talking about species yet. We will come to the next slide. But we have to do some sort of classifications to make more systematic, so that we don't cram our mind with so many, we just simply deal with it, then you can't think. Okay, so how? Two drivers. So we have to start by doing some classifications, so that at the end we know which subgroups, which group and which subgroups are relevant to certain disease. Okay, so that will make things easier. What we say now, we always make things more systematic. Okay, if you as a student, you want to deal with so many costs, okay, you have to be systematic. Otherwise, if you just simply put everything in your heads and then go to the exams, then you got confused. Okay, so we have to somehow play around your mind how to segmentalize these different informations. For molecular biology, how you want to keep in your mind so that they don't get mixed up and make you confused at the end. Same thing we do here in microbiology. We want to classify them for the sake of to simplify, so that it's a lot easier to see. At the end, we want to see the correlations, so that they can lead to a better management. When we want to deal with disease, all about management, how you manage it. And of course, to manage, you need to get information as much as possible. The characteristics, the growth patterns, and the associated disease, so on. Okay, that's why we have books. Okay, we got a lot of books talking about this. And of course, we don't read all. You just find the one that are relevant to specialize and so on. So this is one technique that has been developed earlier, where we are able to classify between gram-positive and gram-negative. And then later on, we found out that there is actually a patterns, common properties, common behavior of these two groups, where we know that gram-positive are more common in the skin infection, in the respiratory area, they are more exposed to the air. And we know that this gram-positive, they are aerobics, they need oxygens, as compared to the gram-negative, where they are more localized into your intestines or away from the oxygen, away from the air. So that's why if you're dealing about gram-negative, we're talking about facultative anaerobes or anaerobes, while gram-positive, they are commonly aerobes. And because of that, they, they are more common in the disease that are related to the exposed area. In the lungs, these are exposed area, while gram-negative in your intestinal infections. Although at the end they can get into your bloodstreams. Those gram-positive, gram-negative can get into the bloodstream and cause a very severe infection, bacteremia and septicemia, probably because the bloods are very rich that allow different group of bacteria to grow, to live, because our bloods are actually very rich in nutrients. Because our bloods are actually the vector, the transporting vector that carry all the nutrients from the stomach to feed all the cells, all the whole bodies. So without blood, we cannot live. Okay, so that's why if there is a saying that actually emphasize for you to take care of. If you have good blood circulations, you can live longer. So, which is, I think, is true, because the bloods are actually the one that are carrying all information, all the nutrients to feed all the cells. So as long as your cells are being fed with the good nutrients, with sufficient nutrients, then you can live longer. Okay, I think I don't elaborate much on this because you have done gram staining. I think you know. Okay, what else? This is the earlier way of doing classification, because at the time they don't have a good microscope yet. They don't have electron microscope. Now we are able to see, with the advancement in the technology, we have an electron microscope. We have what else? There's a lot more advancement in the microscopy. We call it microscopy. There is actually a field just to explore the microscope, where we are able to visualize internal cells, the internal part of the cells, rather than simply by looking on the surface. But it started since a long time ago. They don't have the technology yet. They started with just by looking at the surface. So what they can see, how the color, the characteristics of the colonies, okay? Because they don't have the technology yet. So they look at the color, the shapes, then they have gram staining. They can see the bacilli, the rod shape, and so on. And then with the help of the certain staining, they can differentiate based on the color. We got the differential stainings, gram staining. What else? The staining for mycobacteriums to visualize the spore. Okay, and then make use of the capsules. It's all about staining, to stain the capsules, negative stainings, and so on. So very surface. This is the phenotypic approach, the conventional way of doing it. So now we have microscopes. We have electron microscope where we are able to penetrate to see inside the cells. And in fact, now we are able to see the DNA. Okay? We don't bother how the structure of DNA. We want to know the dynamics of the 4 base of the nucleotides, the dynamics of the composition of the nucleotides. How many A's? There are 4, A, T, G, C. How many A's, how many T, how many C that constitute the whole genomes? I'll give you one example here. Okay. Sometimes there is a good correlation. Previously, the scientists have accumulated data based on the phenotypic formations. Now we are able to see deeps into the cells, and we always do the comparison whether they are consistent or not. Okay, at a certain extent, there are some consistencies whereby, for example, gram-positive. We know that gram-positive, they have a thicker cell walls. Now, with the DNA technologies at DNA, we are able to count, to infer the number of the nucleotide base. How many G, how many C's. Why? Because with the advancement in the chemistry, if you go deep at the molecular level, this chemistry, G will always bind with the C. with 2 bonds or 3 bonds. I can't remember. But there are more bonds connecting G and C as compared to the A and T. So meaning that if you want to break the DNA, remember DNA is a double strength. If you want to amplify, you have to break into a single strength. So you have to put energy to break the bonds. But you need more energy to break G and C because there are more than 1 bonds. While you need less energy to break A and T because there is less bonds. So how we make use of these formations? When we study gram-positive, you want to break up the DNA, you need a certain amount of energy, and there is always a difference between the amount of energy required for gram-positive and gram-negative. Okay, because, gram-positive, gram-negative. Why? Because gram-positive, normally they have higher GC contents in their chromosome, in their genomes. Okay, so that will provide characteristics that give an characteristic for gram-positive. Okay? And it is consistent that they are, when we do compare with the phenotypic approach, there are actually some differences at the phenotypic level where they have a thicker cell walls. But there is another consistency in terms of the DNA makeups where there are a lot more GC. So what are the implications of our work? I have been working in gram-positive for quite a long time. So normally I would say it's a lot more challenging to work with the gram-positive as compared to gram-negative in terms of the molecular work. Because I want to get the DNA, I need to do a more rigorous approach to lyse the cells because they got a thicker cell walls. At the same time, the DNA are more stable because they got higher GC contents with the higher bonds. So to break the DNA is actually not very easy. So to break up the gram-positive cell wall, you need more enzymes, more rigorous enzymes. While to work with gram-negative, you just simply heat up with the minimal enzymes, it will lyse. Okay, and then DNA also can be easily lysed because it got less bonds. Okay, so there is implications. So we can see how this information lead to the consequences by knowing this, how that affects our work as a scientist. So that can be a characteristic of to present the character of gram-positive by looking at the GC contents. Definitely, if it falls within a certain range, then you know this is gram-positive. Not gram-negative, because the gram-negative subtle not have so much, the energy to break up the DNA. And of course, there are other techniques like, I'm not going to discuss on this because it's too advanced, but we will be talking on the 16S, because as an attempt, to classify, okay, to make things more systematic, we need to get a lot of information and we need to correlate that particular trait with certain groups so that we can see the differences. Later on, we can expand, the association can be expanded further. And we have been doing this. Probably this will be a never-ending work because a lot more new information that we might be expecting, and we have to keep expanding the information. Starting from earlier, where they rely much on the phenotypic, now we have the genotypics approach. In fact, now we are able to sequence the whole genomes. We have new technologies in sequencing. Although DNA sequencing has started a long time ago, but we have improved the techniques a lot, where we can sequence a lot faster. Previously, it would take weeks, to sequence the whole bacterial genomes. And to sequence the DNA chromosomes sound possible at the time because it got a lot more complex, a lot longer, our DNAs. But now we are able to sequence human chromosomes faster, and they're a lot faster for bacteria. Within a day, we can get the whole sequence. We can sequence the whole genome of bacteria within a day, and it's more accurate. Definitely with the help of computer systems and so on. So this is huge. Actually, it's a huge scopes where you can explore. So we have like MLST, which I'm not going to talk on this. This will be something that I will discuss in your advanced microbiology later on. But for basic, we just discuss on CTS because this serves as a very basics criteria to do the taxonomy, to develop the taxonomy. What's taxonomy? To correlate all the living cells, how they are being connected. Okay, we are part of the living systems. We can be connected in the system as well. So where are we? We are Homo sapiens. We are here. in the different groups, and then the bacteria in other groups here. These are higher, more complex organism, bacteria, and then the more complex humans, animals. We are more complex, so we are here. The bacteria are here. Okay, how they come up with this? By doing the pairwise alignment of the 16S ribosomal RNA. Why this genes? This gene is a very basic housekeeping genes. It involved in the in the DNA replications. Every living being will have these genes to sustain the DNA systems. So without these genes, probably the cell will die. It cannot sustain. Okay, to keep the cell alive, the DNA will have to be keep replicated and form the new cells. One cell will die, replaced with the new siblings. So that's how we sustain our life. And that's true for microbe, but humans, as far as we live, our cell will die one day. It will be replaced with the new cells. But of course, eventually the whole will die. Eventually, we will die. But at least before we die, how to sustain? We have intact organs. Okay, before you die, maybe you live for 60 or 70 or 80 years old. But during that periods, you'll have to maintain your systems. Okay? So cell will die and replaced with the new cells. So this is one of the examples involved. So it's very important housekeeping gene that exists in all livings. Okay, and yet it does not mutate that aggressively. Okay, gene may mutate, but this gene are not supposed to mutate. There will be some small mutations, but up to the extent that it will not affect the functions. Sometimes the mutation will affect the functions. It's functioning a certain way, but due to the mutation, it's not functioning properly. That's why the cell eventually dies, because it's not functioning properly. But this particular gene is conserved. But there is some small variation that will differentiate the origins. They differentiate your species. Okay, small variation, but not to the extent that it will affect the function. So the function is still similar, but there is some small variation that will tell you your identities, your origins. That also apply to the microbes. We have a gram-negative. What are gram-negative? We have Escherichia coli, Pseudomonas, Salmonella. But we go line the traceability line. It will be connected to a certain, we call it family. Yeah, we just play around with the terms, how to differentiate we come up with a term to differentiate the levels of progressions. Starting from the strains, and then strain belong to the same species, species belong to the same genus, genus belong to the same family, and what else? This is taxonomy. Okay, and this is the line of traceability. Strains, then we have genus, we go from the top. Domain, we have phylum, class, order, family, genus, species, and strains. Okay, normally we don't try to trace back to the domain, because this is not our area. We are more concerned on the isolation identifications for medical purposes. But for biologists, microbiologists who are really exploring microbes, so this is the area. Because I'm a microbiologist, so I was taught on this. But it's good for you to know, although I can see that how are these relevant to your job scopes. You have to know, because this is the background of microbe, but eventually you just focus more on identification and so on. But that's more relevant to your job scopes. But what are the implications? There are implications. Okay, probably I don't discuss it today. Why we want to know the strains, why we want to know the species, why we want to know genus, especially the strains. A lot more issues where we are trying to trace up to the strain levels. Okay, I'll give you an example. But before that will be the last example before I end the class. But before that, let me brief a bit about the concept of taxonomy. The taxonomy started by relying previously, by relying much on the phenotypic approach, where there are a lot of limitations. But that is okay for that time because they don't have the technologies yet. But now we have the technologies, especially with the era in the genomics. We have when we are able to sequence the DNA, make use of information, then we are able to see the more sensitive informations, okay, more accurate. And in fact, but luckily there are some consistency between phenotypics and genetics. There is some consistency. So, but there are some cases where we have to revise, earlier scientists, they are relying on a phenotypic, they have proposed certain names merely based on a phenotypic approach. But later on, when we are able to see at the DNA level, we know that this is not really true. So we have to rename, okay? Because of the new information, we have to revise the taxonomy system that has been established in the past. So this is still ongoing. Perhaps E. coli that we know now probably will have to be renamed in the next 10 years or 20 years. We don't know. But so far, with the information that we have, we know E. coli is good enough. The classification for E. coli disease characteristic, which is good enough, it works. But we don't know in the future, probably we need to revise. That happened to the organism that has been working with the Streptococcus pneumoniae. Before it was named Streptococcus pneumoniae, it was named something else. Okay, but with the advancement with the new data we have, they were renamed. But unfortunately, I forgot what were the original names. It's okay. But at least I let you know that actually what we have established in the past will not last forever. Okay, it may be revised with the new information that we may have, then we need to go back and revise. Okay, this is one of the among the earlier scientists that work on taxonomy. You can read this, a challenge, a lot of challenges. And then with the advances in genomic era, then the taxonomy that we have now is the integration between phenotypic and genotypics. So the way we name it, the one that you see in your manuals, in your textbooks, are only species and genus. And sometimes you can see the strains, the serotypes. So normally you find quite frequently E. coli, Pseudomonas aeruginosa, Salmonella enteritidis, Shigella sonni, what else? A lot more. Those are familiar organisms that are been causing disease all times. That's why we are familiar, because they're very common causing disease, especially the food infection, foodborne disease, food poisoning, Shigella. If you want to work in a restaurant, we have to get vaccine for typhoid fever, Salmonella typhi is spreading throughout the foods as the medium of transmissions. And originally, it's coming from your bodies, from your intestines, but you are not practicing good hygiene, you are spreading on your foods, or probably by touchings. But food, because you are eating foods, so definitely straight away go into your intestines. But if just touching, probably you stay on the skins, then you go and get bath, then you just remove everything. But when you contaminate your foods, somebody else and eat the foods and go straight to the intestine and initiate infections. So normally we will put the genus species. Escherichia coli, Escherichia is genus, coli is the species. Okay, and when you put the bacterial name, you write in italics. Okay, the first time you mentioned it, you write it full. Enterococcus faecalis, for example. But so when you write long essay, you will need to mention many times. You don't put the whole names so many times. First thing, you put the name, you have to spell full. Second time you want to mention, you just put E, just E and faecalis. We don't put Enterococcus anymore. That's the way we do the writings. First time it was mentioned, you have to spell full in italics. Second time, we just put E and faecalis, so that we know that this is Enterococcus faecalis. How about the strains? Okay, strains, go to the strain levels. I'm working on Streptococcus pneumoniae. There are about 100 serotypes. It's all Streptococcus pneumoniae. So we have the genus and species thing. But we are dealing with the strain level, where we have Streptococcus pneumoniae, but different strains reflected by the different serotypes. We have Streptococcus pneumoniae, serotype 19F, Streptococcus pneumoniae, serotypes 7, and we know, based on our epidemiological investigation, we found out that the certain serotypes are more prone for certain level of disease. For example, serotype 19F, serotype 43F, they are very common. They are in your lungs. Somehow they are like pneumophora. But they can, in certain cases, they can lead to disease as well. But there are certain serotypes which are not very common in your lungs. For example, Streptococcus pneumoniae, serotype 7, and serotype 1. If you simply take a take a sputum sample and culture, you may get Streptococcus pneumoniae 19F. It's very rare to get Streptococcus pneumoniae serotype 1. But when you deal with a highly invasive case, so you take blood sample, patients they are dying because of the bacterial infection in the bloodstream, you may get serotype 1s. So somehow there are certain preferences. Certain serotypes, they don't spend time in the lungs. Okay. Once you acquire, they definitely find a way to invade and go to your bloodstream. They don't spend time in the lungs. But certain serotypes, they just stay for a while in the lungs and live as a normal flora. So what makes this difference? That's why that lead to the research. Is all about Streptococcus pneumoniae, but there are some variation at the genomic levels. Probably, okay. Now I go back to the what is actually the strengths. What are actually the information the good information that we can get from when we are able to see at the DNA level, when we understand the whole genomic contents, we are able to see, okay, when we correlate with our naming systems, when we correlate with the taxonomy system, for example, Streptococcus pneumoniae, but there are many serotypes. So when we compare the genome of these different serotypes, they're all Streptococcus pneumoniae. They are sharing up to at least 70%. It could be more. Okay, they're all Streptococcus pneumoniae, and they're all sharing 70% at least similarity in terms of their genomic contents. Okay, and we make it as a criteria. Okay, if you want to classify, if you want to say that these bacteria are actually belonging to the same species, then they will have to share at least 70% similarity, okay, to be claimed as the same species. So how about the other 30%? Okay, that 30% will lead to the strains, the variation at the strain level, where normally this 30% will involve an area or segment of the genome which are easily subjected to mutations. Okay, remember there are housekeeping genes, very important housekeeping gene to maintain the basic life requirements. Without this gene, the cell will die. But there are certain genes which are not actually housekeeping, but they are contributing to the virulence capacity. Without the genes, the bacteria can still survive because they still have the housekeeping gene to maintain the basic life requirements. But when they acquire these particular genes, they become more aggressive, they become more virulent, and they become antimicrobial resistance. Okay, so that area of 30% are the variations that exist, the variation exists and keep mutating. They may be losing or they may be occurring. Okay, so that also explains the strains differences. Okay, as I said, for example, the Streptococcus pneumoniae, there are 100 serotypes, and these differences are residing in that 30%. So that 30% are very interesting to be investigated. Okay, because they are probably different among different strains. So some strains, some serotypes, some strains are more virulent. Why? Because of the differences. How they acquire from other, bacteria, they are living like a community, they are exchanging genetic materials. Okay? Probably they are acquiring genetic material which are present in E. coli and take the fragments and incorporate in the genome and emerge to become a new variant that are more aggressive, which is the same thing what's happening now with the COVIDs. Virus, I find it more, basically, although it's a bit difficult to word virus because you need tools, but actually, viruses are carrying a very small piece of information. The DNA is RNA is very short as compared to bacteria. Bacteria will be thousands of genes, but bacteria is just a few hundreds. So it sounds like easy to deal with it because you're dealing with only a few hundred nucleotides. But of course, to grow a virus, you need to have tools, not as bacteria, where you can, because they grow, they repeat as long as you provide the good media, they will grow. Within a day, they will grow. But virus, you need to maintain the cell culture, which is very sensitive because cell culture doesn't have protection. Human cell doesn't have protection. It's very fragile cells, easily contaminated, easily break up. Bacteria have cell walls that protect the cells. So they are a lot more strong because they got cell wall that protect them. While human cells are just some membranes without cell walls. you can easily damage, you can easily get injured and so on. It grow very slow. When the more time it takes to grow, the more chances of getting contaminated. So that's a challenge. So there is always good and bad things about, doesn't matter whether you work bacteria or virus, there is always challenges that are related to the specific microbes. Okay. I think that's been too much. So next week I'll be talking on the history. So then probably I can finish all slides by next week. So, but I have 2 more sessions with you to go and look into specific issues related to this. So that's why I have uploaded the supplements, the reading supplements. If you want to really see the, when you listen to lecture and when you do the readings, okay, the environment is different. Okay, lecture probably you may miss many things because when I start talking, I will not stop, and you may forget things that I mentioned earlier. But when you do reading, everything is there. You forget and you just go back. So that's why, but it works both. So both complement with one another. That is just the way we learn, by listening to the lecture and sometimes by doing readings. So the reading will help you to to strengthen further, and then to recache point that you might be missing. So, but of course, I don't expect you to. I don't want to complicate you with so many technical things and too many information, because I suppose this will be a very basic, just for you to get you exposed and to know what are the actually things going on and what are the issues, what are the challenges and so on. But definitely there are certain things that you have to know. Okay, there are terms that you have to get familiar. There are some basic information that you have to remember. But I don't think those will be very burdened things for you, because those are very basics. But the challenge is actually to explore, to solve a lot of issues going on. Now we have the pandemic, but actually we have been struggling with so many things. Pandemic will come and go, but all the time we are always facing with the emergence of new variants, problem with the antimicrobial resistance. Why we come up with vaccines, but eventually the vaccine will not be effective anymore. So we have to come up with new vaccines because of the evolving nature of the microorganisms. They evolve. Okay, Streptococcus pneumonia, and true for the other organisms as well, has been studied 100 years back. But some say Streptococcus pneumonia is an old pathogen because it has been studied more than 100 years. But now we are still dealing with it. We have still been dealing with Streptococcus pneumonia. So have not we do much to understand? I think we have done much to understand, but what we have done become obsolete because of the mutations, the emerge. Once we understand, it's probably relevant for a certain period of time, but for the next decade, we will have to revisit, revise, because of the emergence of the new variant. They are carrying a new traits, new characteristics. Okay, we know that Streptococcus pneumonia, why they are named Streptococcus pneumonia, because they are causing pneumonia. But until now, they are still causing pneumonia. Do we have a treatment for that? We do have. Normally, pneumonia will treat with antibiotics, but the problem is antibiotics are no longer effective because they are getting resistance. And then we are also dealing with the clonal spread. Okay, that's why I mentioned about strains. You go to any country, they will have problem with Streptococcus pneumonia. But if you want to think at a strain level, this Streptococcus pneumonia that the Japan is facing, that is Malaysia is facing, are a different Streptococcus pneumonia, or probably a local Streptococcus pneumonia, or probably the same strains. Okay, the same strain that originate from an area, and that same strains has disseminated into many countries. Okay, so that is the questions. I think you can understand this because now we are dealing with the COVIDs. We believe that some say COVID originate from China, although it requires more evidence, but that's the perception, because it started from there. So, and the same COVIDs, COVIDs actually is a popular name. Actually, it's adenovirus. And adenovirus actually present throughout the world. But this one is actually at a strain level. We call it COVIDs. still belong to the adenovirus, but this particular group of these clones, because they are similar, have been spreading throughout the world very fast, which I'm not surprised because this is because they are airborne, because they are spreading through air, so they can easily, transmit. And now we are traveling, humans are traveling from one area to another within a day. You can reach United States. So you can see how fast we are traveling now. And you are carrying your own germs. Okay, so that's why the one of the high risk, one of the risks, okay, if you're talking about disease, is the airborne germs, because they can be easily transmitted through air, and people are traveling that can carry the germs throughout the world within a few days. Okay, so that is a challenge. So I will stop here by giving you the challenges. Then we have to think which that is actually the prospect of your career to solve the problems. Okay, so I want to stop here. It's almost, I don't want to spend 2 hours, it's too long. I think I have delivered the idea of the challenges and why we want to study microbes. Okay, there's a reasons why we have to study this. Any questions? I'm going to stop. That's all for today, and see you this Friday. Any questions? If no question, then thank you very much.
Thank you, Doctor.
Thank
Thank
you.
you, Doctor.
Thank you very much, Doctor.
Thank you, Doctor.
you.